Hi folks, welcome back. And in this video, I want to keep talking about the patterns that we find in the structure of DNA. And today I want to look at how DNA is packaged and stored. And you might recall that when I was discussing the amount of information in DNA, I said that with the exception of the gametes, we have 2 copies of our genome in the nucleus of our cells. 1 copy of our genome is about 3.25 feet long, so that gives us a total of about 6.5 feet of DNA in our cells. But the compartment where it resides in the nucleus is only about 1 to 5 micrometers in size. That's 1 to 5 1/millionths of a meter, or 1 to 5 times 10 to the minus 6 meters. So how does all that DNA fit into the itty-bitty nucleus of each of our cells? Well, that's a great question, and I'm glad you asked it. And now I just hope I can answer it for you. Well, here goes. And what I'm going to review with you is how this works in eukaryotes, or what people call higher order organisms, things like us. So, to overcome this daunting storage dilemma, DNA undergoes excessive levels of packaging and condensing. And to begin with, the DNA first undergoes what's called negative supercoiling into a bead-like structure called the nucleosome. And if you look at this slide, it depicts this nucleosome structure. In humans, a nucleosome consists of about 200 base pairs of DNA wrapped around a protein octamer of small basic proteins called histones. The octamer consists of 2 copies each of 4 different but similar histone proteins, so there's 8 in total. The DNA forms 2 loops around the histone octamer and then goes to the next octamer. And the piece of DNA that's in between the two is called linker DNA. This structure brings regions of DNA that are farther apart linearly into closer proximity. The histones in the octamers are basic proteins, meaning they have positive charges on them. And the DNA has negative charges on it. And guess what? Opposites attract. That's why my wife married me. She's beautiful and wonderful, and I'm, well, not. I mean, just look at me, right? Anyway, the opposite charges attract here in this scenario, so the positive histones have an affinity for the negatively charged DNA, and they stick to one another very well. When DNA is associated with histone proteins, it's called chromatin. And in this first level of coiling, the appearance of the chromatin structure is often referred to as beads on a string, because that's exactly what it looks like. And if you look at this image, you'll see what I'm talking about. This is an electron micrograph of the beads on a string DNA structure. It looks just like beads on a string, doesn't it? Pretty cool, huh? Well, then these beads on a string, that structure gets wound around into what's called the 30 nanometer fiber. And this is shown here. So we're over here now. There's the 30 nanometer fiber. And this gives the chromatin DNA a rope-like appearance. This structure then supercoils into the 300 nanometer structure. And then you have scaffolding proteins that enable the chromatin to undergo more compaction into the visible structures that are seen during metaphase of mitosis. And I'll have more to say about mitosis in the next video. But these structures are sometimes referred to as the 700 nanometer structure, which are divided up into structures called chromosomes. And I'm guessing that you've probably heard of chromosomes. Our genome, all the nuclear DNA that we have, is not just one long strand of DNA. It's divided up into separate pieces, called chromosomes. And chromosomes come in pairs. And we humans have 23 pairs of chromosomes in the nucleus of our cells, or 46 in total. You have 2 copies or pairs of each of your chromosomes. That's because you get 1 of each of your chromosomes from your mother and 1 of each of your chromosomes from your father. Each copy comes from your parents as a complete set of all your 23 chromosomes. But your chromosome pairs are not identical. Each copy has some minor parts that are unique. And in these unique areas, the DNA sequence is different. And this is what is referred to as genetic variation, or polymorphisms is another word for it. And boy, is this an area where the evolutionists love to lie to us and deceive us by making things that have nothing to do with evolution look like evolution, which is why I'm going to address this topic extensively in future videos. But for now, let me address the patterns in design that we find here in the packaging and condensing of the DNA. And once again, we find patterns in the charges in the structure of DNA. So, as I said, DNA has negative charges on its outside. And if you look at this slide, which I've used before, you can see how the alternating deoxyribose and phosphate groups, which make up the backbone of the DNA, would create a structure that's covered in negative charges on the outside. And the negative charges, let me show, they're right there. See that? Here, here. Remember, the bases end up in the middle of the DNA, while the deoxyribose and phosphate groups end up on the outside. So all these negative charges will be on the outside of the DNA. And as I said, the histones that the DNA wraps around have positive charges on their outsides. Those opposite charges attract, enabling them to stick together. So if there isn't a pattern to these charges, and DNA can't wrap around these histone proteins, the fallout would be that the DNA would not be able to be packaged and condensed to fit inside the nucleus of a cell. And if DNA can't fit inside the nucleus of a cell, it would be subject to much more degradation and lose its functionality. It could not be the molecule that it is, and without DNA being the molecule that it is, life cannot happen. And you must know all this, everything involved in the packaging and condensing of the DNA while you're making the nucleotides, before they get incorporated into the DNA, because they're the building blocks of the DNA and they come first. Without the design of the nucleotides and the entire DNA molecule, this would not be possible. So you must know everything about the final product, the DNA, before you're making the building blocks, the nucleotides. Like I've said before, you have to see and know the structure of DNA ahead of time, before it's even made, while you're still at the stage of creating nucleotides. So then, when they get incorporated into DNA, the correct DNA structure will be the result, with the alternating phosphate and deoxyribose groups making up the backbone. So the negative charges from the phosphates will be on the outside of the DNA, so DNA can be attracted to the positively charged histones. So you need to know where to position the phosphate and deoxyribose groups in the nucleotides. So when they create the DNA, they'll end up in the backbone, on the outside of the molecule. Yet again, you have to see and know the structure of DNA ahead of time, before it's even made, while you're still at the stage of creating nucleotides. And yet again, this structure tells us that intelligence and prescience are an absolute requirement for and during the creation of the structure of the nucleotides and the structure of DNA. And once again, you do not get such patterns from randomness. Randomness, which is the basis of evolution, does not and could not create such an organized, logical, intelligent, ordered, structured, and essential pattern of positioning of phosphates and deoxyribose groups on the outside in the structure of the DNA. Randomness, by definition, is the opposite of organization, logic, intelligence, choreography, patterns. Randomness does not result from intelligence and prescience because it's random. Randomness is like repeatedly rolling dice or selecting cards from a deck of playing cards. You never know what you're going to get. It's always going to vary, and there's no intelligence, logic, organization, or pattern to it. Or it's like telling a computer programmer to create a program by randomly selecting the figures that he or she uses. Since we know ahead of time that computer language is a binary code, if the programmer was just randomly entering the figures at their whim, you would never get a functional product. Randomness would result in the negative charges in DNA being found in a random fashion. If the randomness of evolution created DNA, there would be a complete disarray of the position of the phosphates and deoxyribose groups with no structure, order, logic, choreography, or pattern to it. Such a disarray of phosphates and deoxyribose groups would prevent the possibility of an organized, logical, intelligent, choreographed, and essential pattern of negative charges on the outside of DNA. This would result in a change in the nature and composition of the structure of DNA. It would not fit into the nucleus of a cell, and it would destroy its ability to perform its essential functions. The fallout from all this would be that life would not be possible. Therefore, randomness, which is the basis of evolution, does not have the ability to create such an organized, intelligent, logical, ordered, choreographed, and essential pattern of negative charges on the outside of the structure of DNA. And we can make the exact same design inference with the histone molecules that the DNA wraps around. They have to have an abundance of positive charges on the outside of their structure. They have to be designed with a pattern to their charges too, so they end up with the opposite charge that DNA has. And just like I've said with DNA, you have to see and know this with the histones ahead of time. And yet again, this structure tells us that both intelligence and prescience are an absolute requirement for and during the creation of the structure of the histones too. And once again, you do not get such a pattern from randomness. Randomness, which is the basis of evolution, does not and could not create such an organized, logical, intelligent, ordered, choreographed, and essential pattern of charges on the outside of the histones. And let me also say that the packaging and condensing of DNA must also include a process for unpacking it, or you would never be able to get at the DNA for it to perform its functions. Without the ability to fold and condense properly, and then be able to be unwound and unfolded properly, you would never be able to access the essential information, and the regions in the DNA without it being ruined or destroyed. And of course, there's a process for unwrapping it too, which I'll touch on when I address the functions of DNA. But for now, let me point out that all the folding proteins, the histones, the scaffolding proteins, and the capabilities within the structure of DNA that allow it to fold as it does are all needed together at the same time, which is yet again a life-essential example of irreducible complexity. So you must know the structure of DNA and its subunits, the nucleotides, ahead of time in order to plan to strategically, intelligently, and intentionally create and partner the structure of DNA with the histones exactly as needed to design DNA to fold and condense to fit inside the nucleus of a cell and still be able to be accessed when needed without being destroyed or ruined. And I have more to say about the way the DNA is divided up into chromosomes, but in the interest of time, I'll wait and get to that in the next video. So come on back for that one. And if you like what you hear, give me a thumbs up and subscribe to my channel. Thank you very much for watching, and as always, we'll leave the light on for you.